Welcome to this Managing Mastitis session. I'm Tim Brigstock, a Policy Director for RABDF, and I'm delighted that so many of you are joining us. Mastitis, along with lameness, continues to be the top-ranked economic disease that affects dairy herds, and we often forget that. So I'm delighted to be chairing this session on managing mastitis, kindly sponsored by Calibre Control.
For that, Tim. Just a brief introduction really from me. So afternoon everyone, and thanks, as Tim said, for joining us for our webinar on managing mastitis. I'm Ben from Calibre Control, and we specialise in products and systems that provide farms with the means to carry out expert and accurate testing on farm, making sure you can maximise yields and keep a firm hand on the quality of your produce. And our AccuMast plates are one of these products, and they allow you to quickly and accurately diagnose mastitis cases, so you can then select the most effective treatment. We represent Ferrer Diagnostics in the UK, who manufacture AccuMasts, and as Tim's quite rightly said, we're joined by Dr. Rodrigo Bacallo, who is a professor of veterinary medicine at Cornell University and also the founder and CEO of Ferrer. So Rodrigo will talk you through how best to control mastitis on farm, and also how to make the most of the diagnostic tools available. So over to you, Rodrigo.
Good afternoon, everybody. Thanks for the invitation. As I've been told, the majority of the audience are dairy farmers. And so I really enjoy interacting and speaking and conversing with dairy farmers. So this is a fantastic opportunity. So I guess presentation is up, and I will be asking whoever is controlling the presentation to move the slides back and forth. Just a little bit about my background. I'm originally from Brazil. I grew up in a ranch / dairy farm in Brazil. We had cattle and beef cattle and dairy cattle. My passion for veterinary medicine was always linked to agriculture, and went to vet school in Brazil, got my DVM degree there, solely to work with farm animals, food animals, and moved to the US. I managed a dairy farm for a year, and then got accepted into a residency program a very long time ago at Cornell already. Did my residency in ambulatory medicine with a focus on dairy cattle medicine, and went on to do a PhD right after that on epidemiology. And I've been a professor here for 13 years now. And I'm, happy to say that this 2021 is going to be my last year in academia officially. I'm going to be accepting a adjunct professorship in another university, but I'm going to be dedicating to my businesses. And we have a business that we develop diagnostics and mastitis vaccines and other kinds of vaccines. Well, the basis of my research has been on epidemiology of diseases. I've done a lot of work on lameness. I'm sure you guys have heard some of it. So the digital cushion work was all done in my lab, associating the digital cushion with lameness and a body condition score. Moved to microbiology and microbiome, molecular biology slowly, and that's my focus today. I study mastitis and calf diseases, including diarrhea and pneumonia complex. So I'm more interested in infectious disease today because we're develop a focus on either developing diagnostics or developing interventions such as immune modulators, recombinant proteins, and vaccines against those infectious diseases. Next, please. Can somebody move the next slide, please? I don't have the control here. Hello? All right, next. Okay, so I had this slide here. There was, and a slide made to the U.S. dairy industry. In the U.S., we have 9.2 million lactating dairy cows, so the impact is bigger. Mastitis in the US costs $2 billion a year, and mastitis in the UK costs 170 million euros. It is, of course, that if you are a lameness researcher, you're going to say that lameness is more relevant. If you're a mastitis researcher, you're going to say that mastitis is more relevant. They're both important. The problem with mastitis is that the losses are incredibly obvious, right? It's a disease that affects the mammary gland. It ruins the quality of milk. It requires, for some animals, as we're going to see, antimicrobial therapy. It's a painful disease. It's associated with culling, reproductive losses, abortion, mortality, and of course, obviously, immense milk production losses. Next. All right, so on average, we're talking about 225 euros or pounds, sorry, not euros, pounds, per cost per cow per year. The cost in the UK for the industry is 170 million. And it's a really well-studied disease. We have more than 40,000 published scientific articles about the disease. We've been fighting this complex disease, multifactorial disease for a very long time, but it looks like we're stuck in the same place and things are not getting better, despite the immense amount of resources that have been put onto mastitis research. Next. So, the problem with mastitis, the reason why it seems like we're not making progress, is that it is a moving target. We had initially, let's say maybe now 80 years ago, 70 years ago, the contagious causes of mastitis, particularly Streptococcus agalactia and Staphylococcus aureus, were the primary pathogens causing disease. They were incredibly prevalent, ubiquitously present, and caused tremendous damage to the milk quality back in the day. But the adoption of simple things like pre-dip and post-dip and dry cow therapy has allowed us to control particularly Streptococcus agalactia really well. And the somatic cell counts, bulk tank somatic cell counts, have come down from 400,000, 500,000-600,000, and now it's common to see farms that are averaging 90,000 somatic cell counts in the bulk tank, which is fantastic, all because of eradication programs taking care of Streptococcus agalactia and Staph aureus particularly. Now, when you have cows that haven't been challenged with these pathogens that usually cause mild disease, for instance, Streptococcus agalactia, basically the guard, when you're holding your guard up, you're let down the guard of the immunity of the mammary gland. And that opens up room for environmental opportunistic pathogens to invade the mammary gland and cause disease. And that's the case today that Klebsiella, E. coli, Streptococcus uberis, and Streptococcus dysgalactia are the main pathogens causing mastitis today. And we rarely see the Strep agalactia. However, we can't, today we're doing a few things differently. For instance, there are some farms that are not dry treating cows anymore. And when you stop dry treating cows, we're starting to see a slow comeback of Streptococcus agalactia in some of these farms. So it's possible that these contagious pathogens are going to make a comeback at some point. Next slide, please. Let's move on. Another one. Another one. There you go. All right. So what is, herd immunity is it's a term that is on vogue right now because of coronavirus, and we're all trying to achieve herd immunity. But when you talk about the microbiology of a farm, we're talking about the entire challenge or groups of bacteria that are challenging a herd, right? So clinical mastitis, it's at the very tip of the iceberg. That's what we diagnose. Cows are sick with an inflamed mammary gland, altered milk secretion. That's the very top tip of the iceberg. But there's all these other problems that are underlying. You know, the occurrence of contagious pathogens in your herd, how important is that? The subclinical mastitis cases, what's your milking routine? What's the herd hygiene control in cow comfort? What kind of bedding you're using? Are you overcrowding or not the herd? How comfortable are your cows? What is the, you know, the quality of the milking machine, the vacuum, the pulses, and, you know, the milking routine, the milking letdown, all of that is underlying factors that lead to a herd problem that needs to be understood better. Next. All right, now I want to focus, you know, change or shift to the focus of this lecture. We're going to review what are the pathogens that cause mastitis, what are the different causes of mastitis, and how they should be treated differently and why they should be treated differently. So let's dig into treatment of clinical mastitis. Next slide, please. Okay, so this is probably the most important slide that I have on this presentation. If anything, I want you guys to take home this slide. It doesn't matter if I'm in New York, in Texas, or in England, or Japan, China, Brazil, Uruguay, wherever places are milking dairy cows. If I take 100 samples of mastitis milk, there's going to be a very typical breakdown of that milk. About 1/3 of those mastitis samples, if I culture it, I'm not going to identify a single pathogen on it. That's basically what we call, you know, a culture negative sample. Those are very important to identify because those are, you know, a low-hanging fruit. Those are basically cases of mastitis where the pathogen entered the mammary gland and the immune system of the cow, the innate immune system of the cow, was able to react to the infection and resolve the infection. So by the time you milk that cow and you notice, oh, this cow has mastitis, and you collect a milk sample from that cow has already self-cured from the infection. So out of 100 cases of mastitis, 33 cases or more, you know, depending on the farm, this could be as high as 50% of the cases are going to be self-cured. And by the time you collect that sample and you culture it, they're going to be culture negative. No more bacteria. Oh, what should we do about those cases? Nothing. That cow has already self-cured. There's no infection. What she needs is time to resolve the inflammation. Don't do anything. All right, so what about the other 70 cases? The other 70 cases are almost equally going to be divided into what we call gram-negative cases and gram-positive cases. Gram-negative cases are cases that are caused by bacteria that have lipopolysaccharides on their bacterial wall. There are going to be two bacteria that are most important, most commonly found. E. coli, which you know for sure. It's a bacteria present in the feces of the animals. It grows in the bedding. It's an opportunistic pathogen. It causes very severe cases of mastitis. And Klebsiella, which is the same thing. They're found in feces in the environment, causes very severe cases of mastitis. Klebsiella is actually worse. Those cases, particularly E. coli, they activate the immune system in such a way that you don't have to treat them with antibiotics. You don't have to treat E. coli cases with intramammary antibiotics. Klebsiella are different. You may have to treat them with intramammary antibiotics. And the last 33 cases are going to be your gram-positives. Your gram-positives are several, and they can be either environmental opportunistic. So you have several species of Staphylococcus that are environmental and opportunistic. Enterococcus and all the Streptococcus or non-agalactase Streptococcus, particularly Streptococcus uberis and Streptococcus dysgalactide, they're environmental. They're found in feces, they're in the bedding, and so on. Corynebacterium, contagious. These are the easiest ones to control once you know and you start diagnosing and treating them. Streptogalactis, Staph aureus, Mycoplasma prototheca. Okay, these gram-positive pathogens, you either have to treat the animals with antimicrobials, or you have to, depending on the case, for instance, Staph aureus, I recommend culling the animal because it's a contagious and chronic, hard to treat case of mastitis. All right, next slide, let's move forward. Let's discuss this study here. This study has been replicated several times. This is a really good study. Amy Vasquez was a PhD student here at Cornell. I was in her PhD committee. She did tremendous work in one of my clients, actually, that uses our products and protocols. And I'm a consultant for the farm. It's a farm that is milking about 5,000 cows. Really good herd, but challenged. They use recycled composted manure as bedding. 6-row freestall barns, milk 3 times a day, 95 pounds per cow per day average. With all this challenge, they're still making high quality milk. They're selling milk with 100,000 somatic cell counts in the bulk tank, which is for our area is extraordinary. Let's move on. So next slide. All right, so what was the question here? The question is, can we selectively treat cases of clinical mastitis based on the results of culture? And is that approach economically viable? So they basically took all the cases of mastitis and they divided into 2 groups, randomly allocated cows into 2 groups. One group, they would treat all the cases with the Cadillac drug that we have in the US, which is ceftiofur. 1 time a day for 5 days. All cases, regardless of what's causing mastitis, what pathogen was causing mastitis, everybody was treated for 5 days with ceftiofur. The other group, they said, well, we're gonna find the mastitis, collect the milk sample, we're gonna culture it on farm, culture it, the milk, find out what's the cause of mastitis and allocate 1 of 3 outcomes, interventions. If the cow had a non-contagious gram-positive microbe, the vast majority of these microbes were Strep uberis, non-aureus Staphylococcus, and Strep dysgalactia. They would receive the cheapest treatment and fastest treatment on the market, which is basically 1 tube every 12 hours for 1 day. So 2 tubes of antibiotics, 1 in the morning, 1 in the afternoon. If you was treated in the afternoon, it would be 1 in the afternoon, 1 in the next morning. That's it. Simple, cheap, dirty, because we know these bacteria are susceptible to narrow spectrum antibiotics. What about the other ones? Well, if they had a contagious pathogen, in this case it was only Staphylococcus aureus, they were removed from the herd. They had a gram-negative infection, which are mainly Klebsiella and E. coli. They did not receive intramammary therapy. Of course, if the animals were systemically sick, they received palliative care, which is fluid, anti-inflammatories, and so on. Nothing in the mammary gland. Next. Next. All right, so total, we have about 500 cows with mastitis enrolled in this study. Blanket therapy, all animals received 5 tubes of antibiotics. So 243 cows treated 5 days with ceftiofur, 1 tube per day, 50% of the cows. And then the pathogen base had 246 cows, of which only 79 cows, so we're talking only 33% of those cows were treated with antimicrobials. So because they had a gram-positive infection. And 167 cows were not treated at all with antimicrobials. Next. This is just a breakdown of pathogens between groups. There's no difference, you know, so this is, you know, basically very typical, like I told you, no growths and presence of pathogens. Next slide, please. All right, what happens? Clinical days. This is basically how many days the animals were detected with mastitis. On average, 4.5 for the animals that were selectively treated. In other words, the animals where only 33% of them received the antimicrobial therapy. The blanket therapy, where antimicrobials were injected for everybody, they actually took longer, not significantly, but it was a statistical tendency of a p-value of 0.12 or 4.8. Next. And what happened with milk production? No difference at all. What happened to somatic cell? Next. Linear scores? No difference at all. So well, we're seeing the picture here already. And I, as a veterinarian and a consultant, I tell veterinarians and farmers, say, oftentimes doing less intervention to cows results in better results. You're saying, well, but treating everybody just makes sense. They're sick. No, it doesn't. It doesn't when only 33% of the cows benefit from the treatment. So what about the 70% that don't require therapy? Well, you're intervening on them, and the chances that you're causing harm by your intervention is greater than the chances that you're causing any good. Because we already know that there's no good that will come out of intervening in cows that don't have a bacterial infection, for instance. They're culture negative. So yes, sometimes these studies show that treating everybody is worse than treating only the ones that require treatment. Next slide, please. All right, this is interesting here. Now it's mortality, and we have, again, a tendency that the blanket therapy, we had a 5.8% mortality or culling versus 3.7% in the pathogen-based. We killed more cows when we treat everybody. We kill fewer cows when we selectively treat them. It's the opposite of what farmers want to do. We need to intervene. We need to intervene. No, intervene when intervention is warranted. And if it's not, don't intervene. And the cows are going to do better. So this was almost significant. Next slide, please. All right, this is important. What do we call hospitals here? These large farms have whole pens. They're dedicated to animals that are on therapy. So when we pull the cow with mastitis, they go to the hospital pen and they stay there until their milk returns to normal and the residue period of their antimicrobials is over, done. So obviously, if you only treat 33% of the cows, and those are the cows that require therapy, and you're not harming the ones that don't require therapy, they're going to spend fewer days in the hospital pen. They're going to return to their regular pen and faster. And why is this important? Well, it is important because the milk from the hospital pen is not sold. That milk is dumped. Some farms are not even feeding that to dairy cows because of inconsistencies. Although still a lot of farms do feed mastitis milk to calves. Now, that difference of 3 days is enormous, and that's a lot of money in the pocket of dairy farmers. Next. All right, so bottom line is there was no harm done by being selective in our treatments. We decrease antibiotic use by 70% or more, because we use a cheaper drug. Gram-positive microbes, by far, with the exception of Staphylococcus aureus, which require, if you want to try to treat it, you probably want to use a better drug. All these environmental gram-positive, Strep hubris, Strep dysgalactia, Coagulase negative staphylococci, or now called non-aureus staphylococci, they can be treated with narrow spectrum, cheap drugs, not the fancy drugs that regulators don't want us to use. So much less drugs, narrow spectrum drugs, fewer days in the hospital, more milk in the tank, no difference in somatic cell counts from those cows, and actually lower odds of culling in the first 2 months. So all around, it's a strategy that is basically the most cost-effective intervention you can put in a dairy farm today. The return over investment for implementing on-farm culture today is 1 to 13. That's how profitable it is to selectively treat your dairy cows. Next slide, please. All right, so how to implement this process? And that's where we come in with a tool to help implement, and address pathogen by pathogen. Next. Let's do treatment according to each diagnosis. Next. This is a picture of this product that we have created to address this problem. It's called AccuMast, and today we have a version that is called AccuMast Plus. This was created, me as a professor here at Cornell, and the dairy farmer that milks 5,500 cows said, well, we need a solution for, for on-farm culture in a way that we separate the Streptococcus, the Staphylococcus, and the gram-negative bacteria. So this plate basically has 3 selective media. The selectivity of the media is basically done by a combination of active ingredients and chemicals. Media number 1, we created a selective media that only grows gram-negative bacteria. Here you're going to find your 2 most important gram-negative. I try to keep the conversation to what's the most relevant. So E. coli and Klebsiella. There are others, Enterobacter, Serratia, Pseudomonas. These will grow there as well, but the bulk is going to be Klebsiella and E. coli, and it's good to differentiate those 2. And the gram-negatives are only going to grow in section 1, and no growth will happen on 2 and 3 if the infection is, for instance, an E. coli. And then we have another layer of technology that allows us to differentiate the bacteria within the selective media. So if something grows in section 1, it's going to be a gram-negative bacteria. And if it grows in pink coloration, it's E. coli. If it grows in blue coloration, it's Klebsiella. If it grows in yellow coloration or white, it's Pseudomonas and so on. So we created, chromogenic identification based on specific enzymes that were released by the bacteria. And so on media 2 and 3. Next slide, please. Let's talk about the first one. The first one, which can be as high as 50% of your mastitis cases. I tell people, when you start seeing a lot of plates that are culture negative, when you look at a milk sample that you plated and there's no growth at all, no bacterial growth, and there's a lot of those, I keep tell people that this is actually good news. It's good news because of 2 things. First of all, you're doing a good job in identifying all cases of mastitis, including the very mild cases. And number 2, your cows are immunologically competent and they're being able to fight the infection on their own. And by the time you're detecting mastitis, they have already eliminated the infection. So this is not bad at all. Culture negative cases will happen. It happens in at least 30% of all mastitis identified. Could be up to 50 or 55%. And it basically just means that your cows are fighting the infection on their own. And so what do we do for those cases? Nothing. We give cows some time. Oftentimes, if it's just a mild, a few flakes, a few clots and curds of mastitis, of milk, in 24 hours, the milk is cleared again. It will go back to normal. If not, in 48 hours, it will be better. So we know that 95% of these cases self-resolve within 6 days. So they only need time. Next. Now, this is a study that we did. Erica Gunda did her PhD with me here at Cornell. This was part of her PhD thesis. And there were some really interesting things that came out of this study. Next. So basically, we had mastitis in quarters, in healthy quarters. We collected samples. The infection with gram-negative or culture-negative cases, the quarter was either treated with ceftiofur for 5 days or received no antimicrobial therapy. Next. Next. So this is the protocol. Basically, when we identified mastitis for 5 days, we collected samples and treated the treated group with ceftiofur. Next. And then we collected samples for microbiome and culture and qPCR, and clinical score was performed on day 8. Next. And on day 10 and day 14. Next, please. So those culture negative cows that we identified, the cure rate, clinical cure, was exactly the same, of p-value of 0.73, 77 versus 81%. Treatment did not help at all those cows. Next. Next. So this slide is kind of interesting, that shows that on the upper slide, we have linear scores, which is basically a marker of inflammation. Linear scores is basically the log 10 of somatic cell counts. And you can see that it goes up after those animals get an infection, and it stays up for a long time. And these are cases of infection. So inflammation has a lot of momentum, and it takes a long time for it to slow down. Infection, on the other hand, which is illustrated here by the relative abundance of E. coli or Enterobacteriaceae, it goes down a lot faster than inflammation does. And that's why, you know, cows can be challenged sometimes with very low challenging inoculums or amounts. They mount an inflammatory reaction, they fix the infection, but they still have the inflammation to deal with. And that's why you find a lot of these culture negatives. These are cows that self-resolve the infection, but they still have to resolve the inflammation component of the infection. Next. All right, so this is a sort of a literature review. It's not a meta-analysis, but a literature review with decision tree. And basically evaluating several different publications. They concluded that, you know, treating or not treating culture negative or no growths doesn't matter. And about 90 to 95% of cases self-resolve fast. Next. All right, now here's the one that farmers have really trouble not treating, E. coli. So this is how E. coli is gonna look like in the AccuMass culture plate. It's beautiful pink coloration in the gram-negative media. Next. The same study that we discussed, the no growth for culture negative, we also looked at gram-negative infections, including E. coli. So we looked at the impact of treating cows with ceftiofur, which is a good drug against E. coli, it kills E. coli, versus controls. And these are the results. Next. What happened to clinical cure? No difference, p-value of 0.72, and this has been replicated over and over and over. 75% of the controls that did not receive any microbials at all, they were just milked out, self-cured, whereas 70% of the ceftiofur treated had clinical cure. Bacteriological cure was the same again, 85%, actually 5% higher again for the ones who were not treated versus 80% for the ones that were treated. Next. 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 If you look at the literature review, also, you know, several articles have demonstrated this. Studies from New Zealand, studies from Europe, studies from Canada, studies from the US. Basically, you know, there's today there is pretty good agreement that you don't have to treat E. coli with antimicrobials. So what should we do with them? Next. What are we going to do? Intramammary antibiotics are not recommended independently of mastitis score. Now, you're going to have some cases of mastitis from E. coli that the animals are very sick, systemically sick. They're dehydrated, febrile, sick, sometimes even down, not getting up. And do those animals benefit from intramammary antimicrobial therapy? No. It could actually kill them. Let's think about this a little bit. Why are cows so sick when they have E. coli mastitis? They're sick because LPS or endotoxins are being released by the death of E. coli cells. The immune system is crashing over in a fierce fight against E. coli, and the immune system is incredibly competent to kill E. coli. And the death of E. coli is releasing LPS. And LPS is being absorbed systemically and activating the immune system, creating a mass immunologically, and the animals are getting sick because of endotoxemia. So if you put something in the mammary gland that kills more E. coli, you could make animals even more sick, even sicker. So that's why sometimes we see that mortality is actually increased when you treat E. coli. That's why we don't recommend. However, what we do recommend, well, for animals that are very sick, we call that, you know, clinical scores of 3. They're systemically sick. The systemic use, particularly of ceftiofur, is a good thing. It may save some cows, not because it's helping kill the infection in the mammary gland, but because it's killing bacteria that are circulating in their blood, septicemia. So we recommend for animals that are sick with E. coli, not the mild cases, but the very severe cases, that you should treat them with ceftiofur systemically, as well as give them anti-inflammatories, whatever anti-inflammatory systemically that you may choose to use, and lots of fluid, both orally and IV fluids as well, for as long as needed. So palliative care is incredibly important for these cases. Next, please. All right, so moderate cases, score 1 or 2, you don't have to do anything. And severe cases, you treat them with systemic antibiotics, not intramammary, systemic antibiotics injected in the muscle, plus palliative care. Next, please. Next. Next. All right, so next, we already talked about this. Okay, what about Klebsiella? Klebsiella is a little bit different. Klebsiellas are going to show in the culture like this. They're blue in coloration in the gram-negative media. Klebsiellas, I tell people, like, if you cross a Staph aureus with an E. coli, and the result of this, the hybrid that would come out is a Klebsiella, from the point of view of how the disease presents itself. It's not contagious, it's simply environmental, but the cases of Klebsiella, they don't self-resolve as easily as the cases of E. coli do. Cases are chronic. Animals stay infected for a very long period of time. They are often systemically sick, and it's probably the pathogen that causes the highest mortality of all pathogens. So it's a bad microbe. And intramammary therapy for Klebsiella has been recommended by some experts and some research, including our own. So in my protocol, I recommend broad spectrum antimicrobial therapy intramammarially against Klebsiella for, extended therapy, meaning for 5 to 8 days of therapy for Klebsiella. Next, please. So this is just a survival curve from one particular herd, and you can see the red curve is Klebsiella, and the blue curve is E. coli. So they're both killing cows like crazy when they get them, but Klebsiella is worse than E. coli. And it's important to separate Klebsiella from E. coli because the E. coli cows, 90% of the cases are self-resolving. The Klebsiella cases, they're not. Next. Epidemiology, so historically associated with shavings, the shavings are wood shavings bedding, although today it's found in every herd, every kind of bedding. I've worked up a lot of cases in outbreaks in sand-bedded stalls. Klebsiella is eliminated in the manure of infected cows. Almost 90% of the cows have Klebsiella in their feces. Environment is the source of the infection. The perception is that the occurrence of this pathogen is increasing rapidly, maybe because we have better diagnostics now. Common cause is mastitis in compost barns and freestall barns bedded with recycled manure. And outbreaks in sand-bedded freestalls barns are not rare. So nowadays we're finding Klebsiella in every kind of dairy system. Next. This is just an epidemiological study that we did. We actually, the result of this genome sequencing study, we created a very effective vaccine that against Klebsiella and E. coli that we're working towards commercialization now, partnering with a company. But basically, I'm the inventor of the patent, and our company owns the rights, and we're working with a company to try to make it available as soon as possible. But it's an incredibly diverse genetically microbe and causes infections in all kinds of dairy farms. Next, please. I want to leave some time for questions in the end. Next. So this is too complicated. So what do we do with Klebsiella? So severe cases must be treated with systemic broad spectrum antibiotics and supportive therapy. So supportive therapy that I recommend, 2 liters of hypertonic saline, give some calcium, some dextrose, anti-inflammatory, plenty. If the animal is not getting up, you want to provide plenty of water, warm water, so that she can be encouraged to drink as much as possible. Next. Extended intramammary therapy with broad spectrum antibiotics. Yes, the difference between, sorry about the typo on antibiotics there. I'm not very good with picking up typos. So yes, we do recommend treatment with antimicrobials against all cases of Klebsiella, actually. That's the big difference between E. coli. We do recommend broad spectrum intramammary therapy for Klebsiella. Next. Probability of cure, even with treatment, is low. So if you don't treat, you're probably gonna save 35% of the cows long term. If you treat, you're gonna save around 60, 65% of the cows long term. It's a tough bug. It's not an easy microbe. Next. Pseudomonas, you're gonna find Pseudomonas, and sometimes it has this either going to be white colonies or transparent white colonies with a white background. It's a microbe that is rarely found unless you have a water quality problem on a dairy farm, particularly the water that's being used in the milking parlor. It may cause severe problem in some farms, particularly if you have a water quality problem. Other than that, it's just sporadically found. Next. Next, please. All right. So what do? The animals are gonna look very sick, and oftentimes it's gonna be chronic, refractory, not gonna get better. You're either gonna end up culling the cow or drying the quarter, which is something that works. So just stop milking that quarter and let it dry up. The animal is gonna need palliative care, just like the other animals. Next. Have all their gram-negatives. They're all going to behave similarly to either Klebsiella or E. coli or Pseudomonas. We have Serratia, Enterobacter, different kinds of gram-negatives, but they're rare. Next. So same thing, they're going to require systemic palliative care because they're going to be producing endotoxin and lipopolysaccharides the same way. So they require palliative care. Next. All right, now we start getting into the ones that are very common. They're also environmental. Strep uberis is the biggest example, and they're going to grow in the Streptococcus section with dark blue colorations. And all of these gram-positives will require antimicrobial therapy. So Strep uberis is particularly tough to treat. So we need intramammary antimicrobials for these microbes. Next. So, you know, the cure rates are not fantastic, but you can see that with extended therapy, 8 days of therapy, you can end up with about 80% of treatment of cure rate for primiparous and 75% for multiparous animals. If you don't treat, they don't get better. So the thing is, these gram-positive microbes, they are host adapted, which means that they can hide themselves from the immune system and stay in the mammary gland, even in the presence of inflammation. So they're very stealthy. They're good at hiding themselves from the immune system. That's why we need to treat them with antimicrobials. E. coli is the opposite. E. coli is like, it turns on it looks like a Christmas tree inside the mammary gland. And the immune system finds it and kills it and resolves the problem. The animal either dies or gets better. These gram-positive microbes, they're like, you know, hiding from the immune system somehow using different mechanisms. And if you don't treat them with antimicrobials, they don't get better. So that's why it's important to find them and treat them. Otherwise, they accumulate in your herd. Next. All right, the severe cases may cause enough inflammation that you're going to find some clinical scores 3, dehydration, fever, inappetence, that you're also going to need some anti-inflammatory and fluid and all of that. And what do we recommend? The literature says strep uberis, you need extended antimicrobial therapy, 5 to 8 days of intramammary antimicrobials. Systemic antibiotics are not necessary. We don't use them for gram-positive infections. They're not necessary. Next. strep egg, or strep agalactia. Hopefully the UK, you guys in the UK is not, you're not seeing a lot of these cases any longer. Although, like I mentioned, with the practice of not drying off cows, not using dry cow therapy, antimicrobial therapy may allow this pathogen to flare up again in some dairy farms. We're seeing that in the US. One of the largest dairy farms in the US is seeing strep agalactia come back because they stopped using dry cow therapy. So we're working with them on trying to improve their diagnostics to find and treat and call strep agalactia. Next. You know, this, all you have to do is identify them and treat them, segregate them. And for the chronic cases that don't get better, about 5% of the cases are going to become chronic. They need to be removed from the herd. Next.
Rodrigo, we're going to run out of time, so could we keep going? Could you keep going, but speed up a bit?
All right, sounds good.
Okay.
So you're going to find all their straps. They're all going to require antimicrobial therapy. And so, you know, the gram-positive, gram there. So next, let's go down to our Staph aureus and finish it up. Next. Enterococcus, just another pathogen. Intramammary therapy required. Next. Lactococcus can be very hard on some herds, and it's good to identify, treat, and segregate cows. Sometimes it behaves just like Staphylococcus aureus, so can be a very tough pathogen. We're working with the largest herd in Russia, and they have a very big problem with Lactococcus, and they're finding a lot of cases. Next. Next, this is just research that we have done on Lactococcus. The majority of Lactococcus is Lactococcus lactis. Next. And they behave like contagious pathogens in some herds. Find a lot of similar patterns and genetically. Next. Next. And extended therapy, they can be hard to, low cure rates. Systemic antibiotics not necessary. Segregating infected animals might be a good idea. Culling chronics is a great idea. Next. These are non-R Staphylococcus. They're going to show up in different colors. They're very easy to treat. If you find them in clinical cases, just pick your narrow spectrum antibiotics and treat them. Next. 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 The last one is Staph aureus, and we created this system that allows us to detect Staph aureus with nearly 100% accuracy. They're coagulase positive, and the enzyme that releases this pink coloration is basically a coagulase enzyme. So if it's pink, it's because it released chromogens based on the coagulase. What do we do with Staph aureus? I recommend culling of all Staph aureus positive animals. I have worked too many outbreaks in Staph aureus in my life, and they are a nightmare. So I have a zero tolerance against Staph aureus. So identify them and kill the cow. Next. And I think, you know, I'm going to stop here. I covered everything that I wanted to cover. I don't know if I have time for questions, but this is basically everything that I wanted to, the message that I wanted to get across. I think it got across. And I don't know if we have time for questions. And thank you very much for your time.
Thanks very much, Rodrigo. We have got a number of questions, and some are quite detailed. So I think if it's okay with you, we could email those questions on to you, and you could perhaps respond, and then we can take that back to the people who've asked them. But just a couple of questions that have come from a number of people. First of all, when it comes to the actual mastitic pathogens, are they the same in the UK as the US? Have you looked at that?
They are the same. That's what I mentioned. They're the same in China, in Japan, in Russia, in Uruguay, in the UK, and in the US.
Okay.
We work with 20 different countries, and it's unbelievable how mastitis is caused by, you know, a dozen of different pathogens all over the world, and it doesn't change.
Okay, thank you. And then there's a question from Brian, which is quite specific. What about treating with anti-inflammatory drugs like Metacam rather than antimicrobial, especially for gram-negative cases?
Yeah, if cases are severe or clinical score number 3, definitely use anti-inflammatory. Other than that, no.
Okay, and then the final one from Florence, because we are going to run out of time. Are blood clots, bloody milk caused by pathogens or more likely other damage?
More likely other damage. So if it's milk with frank blood or blood clots, it's more traumatic than infectious. But if the milk stinks and it's bloody, it could be Clostridium. So normal looking milk with blood is trauma. Stinky milk with blood is necrotic mastitis, either caused by first case of Staph aureus or complications by Clostridium.
Okay, that's fine. There are a couple of questions from academics, notably from Nottingham University, the mastitis group there, but I think we'll pass those on to you so you can respond direct to them, because I think that would be helpful. But can I, on behalf of everyone on the call, the 125 or so, thank you very much for that really interesting talk. And to thank Calibre Control for their sponsorship of it, and to wish you, Rodrigo, the very best. So thank you very much. We thoroughly enjoyed that.
All right.
Thanks a lot.
Yep.
The next session is at 2 o'clock on making antimicrobial benchmarking work for you. So I hope you've enjoyed that. Thank you very much.